This is part 3. So let's have a look at the technologies themselves, the NGS technologies in this part. You have here a reminder of the different technologies and the companies that are developing them. I would just like to draw your attention here on the fact that we will be discussing not only about the massively parallel sequencing technologies, but also they are still used today, of course, but also about newer sequencing technologies that are called smart or single molecule sequencing, like PacBio, for example. And these revolutionized again the field of NGS genomics. So please keep this in mind as we go, because we will be covering these different ones in the next slides. Now let's start with the Solixat technology that is developed by Illumina, and I'm sure you heard about Illumina because it's maybe one of the most popular ones these days. As I explained before when talking about the bridge PCR, this technology uses the sequencing by synthesis principle and uses a flow cell, which is a support with 8 different lines in which you can put your sample of interest. So the first thing is that the DNA is cleaved into short fragments, and then the small sequences are ligated to adapters, and we will be seeing the whole process of generating libraries later on. And these adapters are small sequences, and the small sequences are used to fix the DNA to the support. And a PCR amplification is conducted then using the fixed strand. So the PCR is performed using that SPS technology, so basically detecting the light that is emitted as the chain is growing, knowing that one base is added at one time. So an image is taken of the slide at each cycle, and the cycle is repeated and images analyzed to determine the DNA sequence. So in fact, when we talk about one technology or one platform, it doesn't mean that there is only one device, but there are several ones with different applications and different outputs. You have here an example of some of the devices that are developed by Illumina, and the differences between them in terms of output range or runtime, for example, as you can see it here. Yeah, here. And I invite you to go to see these differences in the company's websites if you are interested to have more details. So the 454 technology is developed by Roche. I won't be going into the details of the technology because we covered it already in the emulsion PCR part, but I will be just highlighting the fact that reads will be here of different lengths because you have different numbers of bases which will be added at each cycle. And the Roche 454 sequencing technology can sequence much longer reads than Illumina, and this is why that technology was very popular for longer genomes or unknown genomes. So for sequencing whole genomes. You have here also an example of the different outputs according to the devices that are developed by Roche using the 454 technology, so different outputs here, and I would again invite you to go to see the company's website if you are interested to have more details. So what we saw previously for 454 and for Illumina is that they were sequencing multiple reads at once, and this sequencing was read was using optical signals as the bases were added. Solid is another kind of technology that uses a quite different approach because here a ligase is involved and we detect the activity of a DNA ligase rather than the activity of a DNA polymerase. So the sequencing process is called here sequencing by ligation and not sequencing by synthesis. So they use here beads as a sequencing by ligation platform. They use a library of octamers that are that have fluorescently labeled dyes coupled to 4 distinct fluorescent dyes, as you can see it here. So each octamer has a specific D-nucleotide combination, and the rest of the bases are random. So at each cycle, the octamer is fixed, and then its labeled portion is cleaved, and then the sequencing is performed. So this is to give you an example of another kind of technology. The ion torrent uses a completely different technology. It differs from the other ones is that no modified nucleotides or optics are used here. So in fact, the sequencing do not make use of optical signals. Instead, they just exploit the fact that the addition of a DNTP to a DNA sequence will release a proton ion, so an H+ ion, and this will release, so the release of this proton ion will decrease the pH. And the changes in pH allow us to determine if that base and how many thereof were added to the sequence reads. So this technology has been introduced to research and clinical laboratories as a device called personal genomic machine. You might maybe have heard about this one also. So one of the most recent technologies developed now is the SMART technology, which is the PacBio. So SMART stands for Single Molecule Real-Time DNA Sequencing. And this technology, in fact, attracted particularly the interest of researchers because you can sequence very, very long reads, and also because you are not PCR amplifying your template, but rather detecting the activity of a DNA polymerase. And this is really important because a lot of publications recorded the errors that can be introduced in the library by PCR amplification. So here this technology gets rid of that amplification step and produces also longer reads. So this is why the technology is really popular now. And here the DNA polymerase is fixed, in fact, in the bottom of a nanowell, and each incorporated nucleotide will release a signal that will be detected. So the technology is still improving in terms of prices, in terms of cost, in terms of capacity. In 2015, for example, the company launched a new sequencing instrument called the Sequel system, and the capacity was about 7-fold greater than the previous PacBio one. So Nanopore technology is one of the other new technologies that have been developed, and thanks to this Nanopore technology, we might be approaching the NIH goal to decrease the cost for sequencing of an entire genome to $1,000. In this sequencing, there is no need for DNA fragment sharing. So you do not need to fragment your DNA. You do not need any amplification step. You do not need labeled nucleotides or other optical instrumentation. I mean, the classical ones that are used. Here you have only two proteins, one upper protein that is used to make the DNA molecule single-stranded, and a second protein that forms a nanopore in a membrane. And this second protein also contains an adapter molecule that is basically used to reduce the speed of the DNA that is passing into the membrane or into the pore. So this is necessary for a more accurate identification of the DNA strand or the base composition of the DNA. So this technology is really perfect for pathogen surveillance, for metagenomics, for any kind of other application where you would need an ease of use, and you do not have the ability to have huge devices, because some of the technologies are developed in a USB stick, so it would allow you to take the sequencer to the bedside of the patients or directly to the fields if you are working with clinical samples, for example. So you have here one of the devices released, which is the Prometheon, for example, and it is designed to operate up to 48 flow cells at a time, or sequencings at a time. So you can see here how many of these technologies and devices are used today with different outputs in terms of read lengths and of sequencing yields. So I won't be going into the details, but this was just to let you know that you have so many different outputs and ranges in terms of read length and in terms of output range that in order to make sure you decide on which platform to use, you need to make sure that you understand fully the differences between them. So the four main advantages of NGS over classical Sanger sequencing would be first the speed, because NGS is quicker as we saw it than the Sanger sequencing. So first the chemical reaction may be combined with the signal detection, whereas in Sanger sequencing these are two separate processes. One read can be taken at a time in Sanger sequencing, whereas NGS is massively parallel. Second thing would be the cost. Sequencing a human genome now even with the Sanger sequencing, aided by data from known sequence. I mean, the fact that we have the human genome now in our hands, the sequencing a human genome would still cost a lot, whereas sequencing it using Illumina, for example, would allow you, or the Nanopore technology would allow you to have a much reduced cost. You also need to have much less starting material, and you can do more repeats than with Sanger sequencing, and this allows you to have a greater coverage, a higher accuracy, and a sequence reliability. Sorry. So you have here advances in the field of genomics in terms of sequencing cost over the past 10 years, roughly, or 15 years. And you have here important reductions in the cost of genome sequencing, as you can see it here, since mainly the development of NGS technologies. So we know that the sequencing cost of human genome is relatively difficult to estimate, but these kind of estimates are done by the NHGRI, which is the National Human Genome Research Institute. And you have here approximation. You have here the white line showing the hypothetical cost that is reflected by what we call Moore's law, which relies on the doubling of the computer power. And you have in green the cost that is highly reduced thanks to the NGS technologies. So here you have an illustration of the comparison of human genome sequencing methods during the time of the Human Genome Project, and the same thing nowadays in around 2016. So the DNA now still needs to be broken into small pieces, but the sequencing process would be much quicker and will generate millions of sequence reads, and we saw that already. You still need to map them back to a genome, and we will be focusing in the next part, which is part 4, on how to generate this kind of DNA library for sequencing, and in part 5, how to map the reads on the reference genome.